I'm a mechanical engineer, definitely not a single particle physicist or chemist or biologist, as the case may be. So what I want to cover today primarily is about the decisions that we make around how we help our cryo community. So I used how do you choose to go from Workstation to HPC? So just to give you the too long, don't read it version of it, basically we do HPC for all of our cryo workflows. There's very little Workstations left now. Move on from that. So just to give you some context about where I'm from and how long it takes to get here, this is the center of the universe right here. And this will eventually go, I hope. And it'll zoom in. And so this is where I'm from currently, right at the bottom of the planet, as most people think of it. And then, eventually, it takes a lot longer than that video to get here on a plane. So then we land up in here. So the institution I work for is quite a large one in terms of Australian universities. We have campuses all over the world. We have a lot of different partners that we work with on a lot of different instruments. The work that I carry out for the university is primarily about really expensive high-performance instruments, and cryo is one of those. So I work here at the Clayton campus, and within about a mile of my office is the Australian synchrotron. A mile to the other side is CSIRO, which is our national lab for a whole variety of things. I do a lot of machine learning applications at that particular site. So the main group that I work for is called Massive. I wish they hadn't called it that. Everybody sniggers every time I talk about my supercomputer, which is called Massive, and it's not really that massive. It's only 4,000 cores-ish. What makes our cluster and our focus very different is that we pretty much only target people in the characterization space. So I'm not really interested in running general purpose high-performance compute activities on my cluster. Doesn't mean it doesn't happen. That's not our focus. We're really focused on integrative HPC solutions. So what that means is we really want, when you buy an instrument, that we do everything from end to end. So for a researcher, they go to one place and everything happens for them. Go to the next slide. Just to give you an example of the type of researchers that we partner with when we carry out these activities. This is Professor Lithgow. His research area is primarily in diseases that are really, really hard to cure. He's one of our very high performance professors at the institution, and as such, he uses quite a number of different instruments. So if I saw how many people own a Krios here, he has done probably 1/3 of the data that's gone through our Krios. He also uses our new lattice light sheet microscope, which if you haven't seen one of those things, you should check out the videos from Genelia. They're amazing. They do live cell imaging. It's 4-dimensional, very high resolution laser imaging. I've got some videos I'll show at the end. Also uses the synchrotron across the road. Also uses international synchrotrons to look at the proteins that they're working on. And the way that we help them is providing all of the compute, storage, virtual workstations, everything that they need to do their full analysis pipeline for them to produce something that looks like that. For chemists, that means something. For me, it just looks like I've probably designed it incorrectly. One of our mandates to make sure that we really help researchers is that every group that we partner with, we never ever want them to say, here's your CD. How many times have you been to a facility and you take really fantastic data and then they say, here's your USB stick, good luck. I'm diametrically opposed to that, and especially in the cryo space because the data is really big and tricky to deal with. So what we do, I think I have, I'll get back to that. So one of the things that, is there any HPC people in the room? That's always exciting. Oh, there's a few, that scares me. So I'm sort of anti-HPC despite being a HPC admin, in that I want people to actually use my system, and I want them to use my system in a way that they find convenient. And I'm happy to deal with inefficient use so long as they get the answer they want out the other end. So I provide what we call these rich online environments. I think it changes name every week depending on who's giving this talk. And primarily these are really high performance desktops. So I provide a workstation in the cloud. Now that workstation is, for the HPC people, is an actual Slurm job. Right? So it has a time limit, it has resources assigned to it. Depending on if you're my friend or not is whether you get special desktops. So I have run desktops on our V100 nodes. I don't tell people out loud very often that I do that because it's $50,000 worth of hardware that someone's surfing Facebook with. But sometimes that makes sense to do that. So the other groups that we work with pretty strongly are atom probe, atomic force microscopy, cytometry. What's the other big ones that we do? We do a lot of light microscopy stuff. So the workflow that we try and do for every instrument is that, I love this picture of the microscope here, is that we go from the microscope into the PC and then directly into a storage system of some sort. So we have two storage systems for cryo. We have an archiving space, which is a web front end, and if you've used Figshare before, basically it's Figshare except it's tuned to deal with really monster amounts of data. That application that does that also pushes it to this very dodgy picture of a cluster, where people come into my cluster through a whole variety of mechanisms, and mostly in the computational biology space, they come in through the desktops. They don't come in through the SSH login nodes very much until they get very experienced. Let's get rid of that. Now, I've probably got enough people here, I can talk through this really quickly. So for creating the samples that they need for cryo-electron microscopy is a very long and time-consuming lab process, which every group I work with, typically nearly all of their time is spent doing that. Where I come in and help is from the data collection all the way through to the model process here. So I'm going to show a better picture of what that looks like. Now, cryo-EM, when I came to this community, it was very confusing to me because I've got a medical imaging background and heavy engineering, so doing simulations and things. So the first thing that, when I saw imagery that came out of this, I asked directly, is the machine broken? Because the images that come out of this thing are basically uninterpretable by humans. So then that's when I went back and had a look at what it actually is. So the processing itself is a combination between computer tomography and photogrammetry. So computer tomography, anybody had a CT before? Have you done the annoying calculations that you need to do to calculate all the back propagation? Really hard. When you use research CTs, you have to do it yourself. It's horrible. So for a medical CT, they take about 1,000 projections. So that's 1,000 different angles, whether you know exactly what you're looking at. When we do cryo-electron microscopy, you have no idea which direction you're looking at. You don't even know whether you're looking at the same thing or different conformations. There's a lot of things that make it a lot more challenging than doing that. The other technique that I'm talking about here is photogrammetry, where you're actually, photogrammetry is where you combine multiple two-dimensional images into a three-dimensional image, where the view that you have is arbitrary. So in cryo, every view that you have is arbitrary, except if you have a particular molecule that tends to rotate to find a surface. I'm not going to talk about those really annoying hard problems today because they make researchers cry. We're going to go, yep, one too many, go back. All right, so what pops out of the microscope is images that look like this. Now, do you think your microscope's broken? If you're used to seeing really beautiful color images from other microscopes, these things look weird. Even if you've done scanning electron microscopy before, these things look like you've done something wrong. If we zoom into them, you can see I've actually picked a molecule that makes it easy to see, well, relatively easy to see. So you can see here, this here is a particular end-on view, hopefully, of the molecule that I'll show the reconstruction of. So this is a C9 protein as part of your immune system. That when you figure out all of the different particles that you think you have, these are the groups of views that you think you've got. This one's nice because you can actually see it, and it's nice and symmetric. I showed this one because it's friendly. And eventually you'll end up something that looks like this. So this is what happens with all the proteins in your immune system. This one is missing 4 of the other proteins that you have in a normal person, but this is most of them. And this is what happens, they all form up, and then this bottom part down here tends to eat through whatever your immune system doesn't like. There's a really amazing construction for this for the Zika virus. If you get a chance to go and have a look at it, that was one of the first indications they knew what Zika virus was once they imaged it, but not this one. All right. So what are the scientists looking for when it's out of this? They are trying to get to this, a reconstruction. So I'll show you what this looks like, assuming I don't fall off the stage. So we can drive this. Where's my mouse? So they're hoping to put into a protein data bank whatever structure they solve, and they put down all this particular information down here. What they get out at the end is something that looks like this. Now, can I turn off polymer? So they get an electron density map that looks something like this. So then they can see exactly where all of the molecules are that they're interested in this particular case. By the way, I really love this website. It is amazing. I actually tried to figure out how to pull that plug-in out so that I could use it myself, but they've buried it inconveniently. So the applications I'm going to talk about today, I've got some real examples. So one of the groups I partner with, and they work on hospital-borne diseases pretty much. So they work on VRE and MRSA, both of which, if you get, are horrible and very difficult to treat. They've published some really amazing papers in the last year and a bit. Just to give you some context about how important the cryospace is, one of the other groups I work with, they published 4 Nature papers in the last 6 months. So now all of the data in there is cryo and a few other things. All right. Now, I haven't updated this slide since the last time I gave it, apart from gave it some colors and modified it. So, of the people who process the cryo data here, how accurate do you reckon the numbers are? Do you think you're processing a lot faster than this? Because this is our average time. So I actually checked with our lab to find out how long do people take when they're going through this process. What is tricky down here is how long it actually takes for them to do the compute. It's a really, really long time. And they go round and round and round. I was having this discussion last night. This particular application has so much human input into it, you can't make it into a workflow, really. The workflow is human clicking the button and making a choices about how they process the next step. That's really challenging for me if I'm wearing my HPC admin hat. How do I make that go fast? That's really hard. So, all right. So because this is a GPU conference, we're going to talk a lot about GPUs today. And part of the reason I want to talk about them too is because it makes some of these parts of the workflow very, very quick compared to the CPU versions. So GPUs are used in the pre-processing step. I've been actually focused a lot more on the pre-processing steps recently just because we're getting more experienced with how we're running the system and how we're running the microscope. So both of those things mean the better pre-processing I can do, the better outcome at the other end, so the higher resolution data. As it turns out, if I give a data set to a beginning researcher, they're probably going to be 0.4 angstrom worse than someone who knows what they're doing, who processes exactly the same set. That's a big difference. That's a difference between getting published in Nature and getting published in the, I don't know, the Journal of Cornflake Packets or something. Basically you either get down to that level of resolution or the stuff that you're doing is not very useful. So beam correction, CTF estimation, particle extraction, all of those, let's go back, are now GPU accelerated to the point that they're quite efficient. I mean, they're still pretty clunky. They still crash pretty regularly. They're not great with bad data. And researchers don't like being told that it's their data that's crashing the system and not the program. I really love having that conversation, if you've noticed. So getting back to the analysis of this, the challenge with working through this data is you don't know very much about the system. So you have to try and aggregate the information you have into a form where you can make choices about what to do next. So I love this quote from Schurz. Go back here. Like, it's complicated by the lack of information, and then you've got structural variability. So basically, something might pop out the other end. So the major analysis steps, the ones that I care about as a HPC admin, are the 2D classification, 3D classification, and refinement. These are really long. They can be really long. We helped a research group that did some 2D classification, and it took 2 weeks to do 25 iterations on a DGX1. So they're a really big problem. It would have taken about 8 months, I think, on my cluster to run that job. I was actually running the jobs in parallel so I could see how fast it was. Already talked about that. I don't want to talk about that. So if we talk about one of the GPU accelerated parts that they're still working really, really hard at, it's particle extraction. This is hard because you've got to try and extract what you think is good data, which is this, out of this very low signal to noise view. It's really hard. How many people are using machine learning particle pickers now? Or you don't know? Maybe-ish. How many are using CryoSpark? You're using CryoSpark? That's interesting. Okay. I saw at least 5 or 6. So I find that interesting. So this space has probably been going 2 or 3 years, and it's really slow compared to other image processing areas. And for me, I'm not sure why that is. It's probably because the algorithms are really hard to perform on such noisy samples. So what eventually pops out after you've made all of your guesses, or you're really lucky and you've got enough grad students or undergrad students that have clicked maniacally on all of the particles, is that you'll be able to sort them into something that looks like this. This is a lot more than most people classify typically. Typically they only classify into sort of 20 or 30 groups or something like that. This is a lot more. There is some advantage in doing it this way, in that then it's a lot easier for you to kill off particles that you think aren't working very well, like this one down here. I'm going to talk about that. All right. What I want to do is cover an example now. So I did ask permission before I used this photo too. This is my friend Matt. Dr. Belousov is a biochemist not sure he'd like me describing him like that, but anyway. And he does a lot of cryo-imaging work with us. This is him in hospital after he got MRSA. He's a jiu-jitsu practitioner, and they seem to have a magnet for getting MRSA for some reason. So this is him when he's about to get treated in the hospital. And you can see he's pretty excited about getting a huge dose of vancomycin or linolazid. I can't remember which one he got for that. So his research work is looking at drug targets for MRSA and the VRE. And the way that they work, and I like how I've borrowed these slides from Matt, he uses a pragmatic approach where they extract the sample from the patient, they then go to the lab, pull out the protein that they're interested in. They also do some gene sequencing, which I'm not going to talk about. Once they've got the ribosomes isolated, that's when they do the imaging. Now, for this particular case, they have molecules that look somewhat like this in different versions that come through the lens. They're also using the faceplate on our system. Do you guys have faceplates? Because if you don't, check out the faceplate data. It makes such a huge difference to your work. It's amazing. I can't believe that anybody doesn't have them. From there, you can see his data is very good. You can see here that's nice and clear. You can see where all the particles are. This is an amazing micrograph. When I say amazing, I mean I've probably seen a couple of thousand of them, and this is one of the only ones where you can see that there's actual particles in there. And then there was a huge amount of particle classes generated out of this. So in this particular example, there was 500,000 particles collected, which is a lot. Depending on the thing that you're looking at. I helped someone from University of New South Wales, and they had 7,000 particles total. So the thing that they were trying to collect was extremely difficult to extract. Unfortunately, their data is sort of awful, so they've got to work on that. What Matt did then is we do a 3D reconstruction. So that 3D reconstruction is based on a guess of what that particle is. Sometimes that's what do you call it? A very informed guess. Other times the ab initio models. Most of the people that I help are actually doing systems that they know approximately what the model will be. And so that works really well. If you're working in a facility where people are scanning things where they don't really know what they're looking at, those ab initio models are very tricky to do well. Actually, I'm going to show. What I'll do is I'll show you a video in a couple of minutes too to show you how the drug target fits in. So the point of the work for Matt's group is to figure out where the drug target fits inside here, which is pretty tricky. All right, so I gave a talk on this about 2 years ago, and I thought, I better review where we're up to now, because hopefully I'm 2 years smarter than I was before, which is not always the case. So in the past, we're probably limited to about 1 to 4 terabytes, smaller numbers of files. What was really important here was cores. So a lot of tools were CPU driven at that point. The GPU versions weren't great. It was typically a lot cheaper and more cost effective to use cores. Now, this year, data sets are significantly bigger. I've got new cameras, so this was probably, there was K1 cameras, maybe K2 might have just come out on this. This is sort of K2, K3 cameras and other things. So we're getting some pretty big samples now. There's a lot of automated sample collection as well, so these things are collecting overnight. GPU memory request has gone up a lot. So the box size that people are looking at is a lot larger. The resolution of the images is much greater. This is actually a significant issue. I'm not sure, we haven't been paying too much attention to it in the cryo community. what's interesting is when you see researchers contact developers of the particular applications in this space, they just say, bin your data, make it smaller. And I'm like, oh, I just went and flew to Japan to use a $10 million instrument and collect the highest resolution images I can get. Why do I want to make them look worse? So we're still struggling a little bit there. Dramatically drop the number of cores that need to be used, at least half, if not a third. That's a big difference. But it does make me think very clearly about what I'm doing next when I buy my next hardware, because it means I need more GPUs and less cores, which tends to make my HPC people grumpy. The other really strong thing that's come out is high IOPS storage is really necessary in this space. It makes a dramatic difference to the processing times. So if you ask politely if you can borrow someone's DGX, copy your data on there and have a go at it, and just see how much of a difference it makes having really high-speed disk locally. It's just a huge difference. Probably not going to talk about that. All right, so I wanted to show you a video of one of the samples that we're going to talk about processing. So this is a really small molecule. It's only 200 kilodalton. This is a very strange processing step. So this one is a fluke in that only 4 hours of beam time was used to collect this data set. They had 3 days booked. They only needed 4 hours. And the reason why they only needed 4 hours was because they were processing on the fly. So they're using the system to process the data as they were collecting. So this particular one, you can see the resolution here is 2.6 angstrom. It's really high, and only 32,000 particles used. I actually use this information in this video to, shame's too strong a word, encourage people who come along and say, I'm struggling to process my 500,000 particles to something, because then you just show this, which was done with 32,000 particles. Hopefully it'll go. So this is what it landed up being at the end. You can see it's very nicely resolved. You can see the hole down the center of the particle. And here you can see how closely the 3D map that was generated to analyze this fits almost perfectly. This one is amazing. So if we remember back to my earlier slide where I said it took 3 months to analyze, this one took 9 days. So why is this one so much different? So this one was processed by I would argue, probably one, if not the number one person that I know of in this space, but he's pretty well up there. So this is what happens when you have super experienced people doing the analysis. Right? It's very quick. Right? Each of these dots here are basically jobs that were carried out. So this is taken from the Relion folder creation. So this is an extrapolation from how they created the files, so I can see what they're doing because I didn't have access to their system at that particular time. So of those really long time-consuming steps, classification was done twice. Yeah, this is really interesting. So in a lot of analysis, they're only done once here. People go through and they go, I know what my particle is, and they pull it out. Well, what this one did was, I went, well, I now know what I'm looking for. How about I go back and I reprocess my particles and go and find all the rest of the particles that I know now what I'm looking for. So being able to perform this step again in a timely manner is really important. All right, so I'm going to talk a little bit now about the GPU architecture and does it actually make a difference. This is a really interesting topic to me because I have some tools that it doesn't matter what GPU I run on, they run about the same. So they're limited by my storage or by the CPU, which is strange to me because the thing's running on the GPU. For Relion, which these ones are based on, you can see there's a dramatic performance improvements when we move to the Voltas. We originally had a P100 DGX, which then became a V100 DGX. You can see here it made a very big difference to us with how fast we could actually process. If you've got a heterogeneous system and you've got cryo people, it's about 2 times faster to go to Pascal as it is over Kepler, and it's about 2 times faster again from Pascal to Volta. So you're getting at least a 4 times speed up with doing nothing, right, apart from just running it differently. When you recompile, it's actually faster again. I find it really interesting for Relion that they provide some strange advice about how to run jobs on particular clusters. If you haven't already, I would sincerely suggest that you talk to your HPC admins about helping you figure out what the most appropriate way to run on their system is, because it's very different depending on what's under the hood. The important thing to note here is that once you hit about the number of GPUs versus ranks for the DGX 100s, basically your performance doesn't change. It's really interesting because in the past the GPUs weren't fed quick enough, or the workload wasn't enough for the GPUs to keep them busy. Whereas now, especially if you've got high-speed storage attached to it, they're very busy. And that's a very efficient way of running. Because then the CPUs can be left for doing other things. So if you've got a parallel file system, for example. I don't remember why I put this slide. So the reason why I put this here is, has anybody got access to a K3 yet? Nope. I know there's not too many of them around the planet. They're collecting 5K images now. I was speaking to a professor from Peking University last night about it, and they're struggling because these images are enormous. It makes it very difficult. So when we started out on this journey, we were down here where we were limited to about 500 box size. Can now go up to just under 600. This makes a big difference for very heavy particles. What I've got now is the 32 gig ones, this will be very interesting. So in the disease space, we'll be able to process very big particles, which I have about 3 or 4 groups that are using CPU only stuff to do those currently. That's very slow. And I feel bad that I can't help them. So if people in the cryo space really love their workstations. I mean, they really love them. They want to buy lots of them and put them under their desk. This is the chart I show them now. As I say, look at the difference between what I can provide you in my data center with access to very high-speed storage compared to what you're going to do on your desk. This makes a huge difference. It's like 40% faster without even trying. It's really important. I'll show another one here. This is a huge molecule. This is 3.8 megadalton. This is a molecule that they were doing targeted drug delivery. So they were modifying a particular MRSA drug, I think it was MRSA drug, to fit into a particular location. And you can see here, that's the drug. So they've actually got imagery of the drug in situ. And so they did this multiple times so that drug target actually worked. It's pretty awesome. All right, so this one took much longer, 14 days. Still a lot less than the 3 months that most people take. So it's still fast. And this one's quite different too, because you can see that there's multiple steps all the way along here. And you can also see a parallel step here where they tried to run 2 models at once. That's something you can't do in your workstation really easily. Don't need to talk about that. So of the 14 days, 6 days supposedly were spent with compute. This is how long it took to create all the files in these particular folders. And you can see some of them take a really long time, some of them not so much. But there was 3 days worth of compute in here that I can measure. That's a lot of compute time. And how do I make that go faster? I was going to explain why that peak's there so we can ignore it. That peak is the particle extraction step. So because this particular guy really knows what he's doing, repeated the particle extraction step once they've figured out what they were actually looking for. Probably something we should do a lot more often. So when I talk about the really long steps, I talk about class 2D. This one confused me. I thought, why is your 2D classification step so short compared to all of the rest of them here? And it's purely because of the pre-processing of the data. Really good pre-processing means that the really hard steps are much more efficient. If this had been done poorly and there was 1 million particles in there, for example, that would take days, and it'd be up here somewhere. All right, so we change the zoom. So that took about 2 hours to do. So that's enough time that you can wander off and get distracted and go and irritate students in the lab. But then there's a big gap between you and your next step. Right? So what I want to do is I don't want them to get up from their desk, preferably, or get up from wherever they're processing these things. Class D 3D step here is really long. And you can see here that the 2 models are generated. And this here. So these jobs only appear for people who know what they're doing, which are refinements. How many people, have you done any refinements of your data yet? So for your publishing? These take a really long time because they're doing the most computationally expensive method they can to create the best result that you can get. They're long. All right. So I want to talk about the 3 main options that we have that people do. One, we have Workstation. So the data I'm going to show is from Matt's Workstation, which is about 8 cores, 64 gig of RAM, 4 gamer cards. It's pretty quick, pretty quick. The things that I provide in terms of cloud HPC, so I should clarify that my HPC is on private cloud, and these are the boxes that we have for that. So there's 3 V100s, a lot of RAM, a lot of cores. These are Skylake, which make a big difference actually. And then we also got some data for the DGX1Vs. So we now have 6 or 7 of these, specifically for image processing applications, of which Cryo is a big one. So how much faster can I go? So if you focus on the super duper fast one first, so this is what happens when we use a DGX, right? Without doing any tuning whatsoever, just taking exactly what the researcher did and then running it on a DGX, I'm now 4 times faster, right? So that's half an hour. I can probably sit for half an hour. I can probably stay for half an hour. These next 4 up here are how we run on the cluster. So depending on how busy our cluster is whether this makes sense or not. So you can see our standard GPU node is still almost twice as fast as the Workstation. What's really important about that is because this community doesn't need compute all the time, it means I can share. So I've got about 40 groups using my system. So then they can all fit in there in a timely manner. I do provide them with enough resources so they get real-time access, at least at the beginning of their processing. And the reason why I do that is because I know it's so much faster to do it this way. As you can see here, as you go up more nodes, the performance doesn't improve. So there is a limitation in a clustered environment about communication across the cluster. All right, just to show the difference for the communication here, these are all the changes that the Relion developers say that you should make. So you can turn MPI on and off. There's a few other things that you can do to supposedly improve it. As it turns out, on a properly designed HPC system, none of these things make any difference. Nothing at all. I thought I'd done something wrong and then I went and checked. But no, it just means that if you run on a single node, everything works as efficiently as possible. Because then what we get is when we run on 4 nodes and you change the parameters, you get things changing here. Right? So the really important aspect to the multi-node is you've got to make sure that the MPI is done correctly, and that you've got a parallel disk file system, whatever that looks like. Mine's Lustre, could be BGFS, could be whatever you want, but it does need to be able to deal with. So the green ones here are 4 nodes, so they will do 130 parallel reads at the same time. And they'll do 150 plus megasecond per channel of that read, so you need something that can deal with that. Actually, that's something for my next cluster. I need better storage. So if we want to have, I wasn't sure whether I was going to put this slide up here, because people always argue about the cost and things in this space. So one GPU node is 1 2/3 faster than a performance desktop. Right? So to give you a rough idea, so the desktop that was used when I showed the numbers was about $7,000 worth. These GPU nodes are about $35,000 worth. A DGX one to me is about $200,000, $250,000-ish $1,000. So you can see here, if we only look at the cost ratio, right, it's 30 times more expensive to have a DGX than the workstation under the desk. That's a lot of workstations I can buy. Right? Why this doesn't work though is each of those workstations needs their own storage. They need their own backup solutions. They need 10 gig pipes to their workstations from wherever they collected the data, because they didn't take it on USB sticks because that's evil. So if you've got a big enough community, doing these GPU nodes or the DGX ones really, really makes sense, right? Especially when you're 4 times faster without trying. I tried to be a bit sneaky here to collect some data out of the Relion developers. So they give you a tutorial, and they mumble some numbers in there about how quickly it should take you to do the tutorial. I thought, that's really interesting. I'm going to see if I can pull apart your tutorial data to see whether I can figure out how fast you went. And as it turns out, they strip the data set so you can't see it. So you only get the last iteration, which is really annoying. So the types of systems that researchers look at, and I still occasionally help people buy these because they're either not on campus or they're somewhere else. These things are expensive. And practically, if it was me buying it and I was doing a couple a year, I'd be probably looking at this. That'd be probably good. That storage is completely inadequate though. I'd have to do something about that. These are really nice because people buy them. They turn up with software on them and they're supported. They come with software. If none of you are really HPC admins, then this slide's sort of useless. But for me, when I look at the system, why they're attractive is because they install all of this software, which if you've tried installing it yourself can be a real pain. Yeah, we talk about research grade software. This is right in that classification. It's tricky. And then you've got all of the sysadmin type things that you need to make sure are compiled correctly and configured, which aren't 5 minutes work for most people. More than 5 minutes work for me too. Just ignore that. So people continue to ask me, yeah, why don't you do this in the cloud? And I say, well, it's really simple. Because at $25 an hour, that's a significant cost. So how much, if we go back to the example I showed with the guy who's probably the fastest processor that I know, that's $8,400. It would cost in compute alone. That's not the storage. Right? So no storage included. So you'd have to figure out some magical way of getting the storage into this thing. Which then gives me this. Whoosh, right? Every time you process a sample, that's how much money you'd be burning through. It'd be fantastic, right? $8,000. That's one of the fastest chemical reactions on the planet. That's why I chose that one. All right, getting close to the end. So what's changed in the last 2 years? So this is what my thoughts were about The pros and cons for Workstation, Cloud, and HPC. Well, it's like, oh yeah, and when I went and thought about it for this year, I go, well, what's really changed? And the really important thing is, realize that the cloud scales. Yeah, there's no problem with it scaling. It does not scale easily. If you want to win in this space, you've got to figure out how you do multi-node things, and that's not easy. Even running my own private cloud, I've got lots of complexity in there. What has also changed is for HPC, the new nodes that I've put in and the ones that I suggest to people, they're purpose designed for this. Right? They're really, really targeted at image processing. So all of the things that can make you go faster, those are the things that we bought. And if anything, I had to rein in some of those things because the HPC people were angry that I was spending so much money on things that were image processing and not solving VASP or Chromax or whatever other thing that they're interested in. The other huge thing for us is containers. Right? So pretty much any tricky piece of software now goes into a container straight up. And I'm about this close to being 100% containers and just not installing anything on the HPC system. I can't remember, we're running about 700 pieces of software and probably 300 of them are containerized now. Makes a big difference, especially when I can give them to the people who've bought the workstation and say, here's a container, this works. I probably don't want to talk about that anymore. So what I looked at in 2017 was most of my cluster was K80s, and I go, whoa, I've got a P100 DGX. Right? How much faster is it? And I'm like, wow, that's great. It's a lot faster. Should do more of that. When I went back and redid these results, I thought I almost wanted to not put this slide up there. I was like, wait a minute, why am I slower now? I've got a new architecture. I've got Volta. I've got newer chips. I've got newer disks. What's going on here? When I went and looked back at this is entirely due to new software. This was an aberration that actually made a mistake. So the software wasn't calculating correctly. So that particular version I had wasn't great. This is the new Relion 3. And this particular example is small too. It doesn't really leverage the DGX as much as you could do. When we had the task, it was important to look at the task to make sure the GPUs were loaded correctly. This has changed over time. So the V100 now is still faster again, which is great. And it's faster sooner here. Yeah, is that the right button? No, wrong button, that one. All right, and just because I can, this is what happens when you've got heaps of them. All right, so that's just over $1 million worth of hardware. So instead of it taking 40 minutes, it goes down to 10 minutes. All right? That's really useful. If you can get a first iteration pass in 10 minutes while you're at the microscope, that will significantly change how you collect. Hooray, like that. All right, what stayed the same? Software dependencies are still horrible. I posted this all the time, I can see. I will make it quick. I love this. This version of OpenMPI is completely deprecated. It's got bugs in it like you wouldn't believe. And the developer really loves it. You can't actually install it from source, from the package, which is really helpful. It's like, that's okay. How do I do that? Badly. Do I want to cover any of that? No. So what I wanted to show here is how hard Relion drives your system. So if you're a HPC admin, this should make you feel happier about how they drive it. So this is 34 cores. You can see it absolutely smashes them. Keeps them pegged. What's it doing though is important here. So this is the reason why we want to look at this space a bit better and why you want to put purpose-built hardware in, because all of this system here is where you need to make the choices. Keep going. GPU utilization looks exactly the same as the CPU utilization. This is something I want to go back to the developers about, because that means that they're not pulling the data in and out asynchronously. So there's some steps in there that mean that this gets slower. Right. Uses all of the RAM is the takeaway from this slide. So however much RAM you can get on your GPU, these things will fill it and keep it full the entire time. Really important. I'm not going to talk about that. It's probably... so probably the things I wanted to cover at the end. So the things that I really see as the things that we need to push into in this space is machine learning and particle picking, is ab initio modeling, so you can do it very, very quickly. And the final thing is improving our pre-processing to the point that for users, they get completely pre-processed data. Practically, I don't really want to give them the raw frames anymore. What I want to give them is the motion-corrected stacks, and then they can go off and do the analysis. And once they get to the point that they're ready for the movies, then we can look at what that looks like. Okay. That's probably it. Oh, I do want to show this video. So this is the last group I'm working with. So we were talking about lattice light sheet microscopy. So this particular here is mitochondria escaping when the cell dies. So this is live imagery that they took. Have a guess how big that data set is. It's 40 TB. Yeah. That acquisition was a couple of hours. Yeah. I'd really like to thank all of the people who contributed to my talk too. Matt Belasoff, who was fantastic. He provided me a whole heap of help. And I'd really like to thank my funders.